Thank you, John, and thank you, Annie. I've learned a few tricks there for sure. And yeah, happy to be talking and sort of give a little bit of an overview of both what Mushroom Observer has been and where we're going, and give you just sort of some flavor. And at the end, very happy to answer questions and so forth. Let me go ahead and share that screen. Great. Okay. So John did a wonderful job introducing me. Thank you so much for that. As John sort of described, I consider myself a naturalist and a mycologist, creator of Mushroom Observer, and currently for my day job, I'm the director of software engineering at a company called IQVIA that helps with running clinical trials. So it has nothing to do with fungi and takes a lot of my energy currently, but we did help get the vaccines approved for COVID, so I feel pretty good about that. So Mushroom Observer, if you haven't seen it, looks like this. Roughly, this was someday a long time ago when I grabbed it, and I see a hand up. Someone wanted to ask a question? No. Douglas Miller, you have your hand up, just so you're aware. Mushroom Observer, as John mentioned, was created in 2006. I had always wanted somewhere to talk about mushrooms and actually make some of my images available and things like that. So this was the very early days of Web 2.0, and I discovered sort of how to do this, and spent a few month or so working on things, and managed to get something that was showing, it kind of did what I wanted it to do in terms of making my images available and giving people the opportunity to upload other images. I shared it with some friends, they shared it with some friends. Many years later now, it's grown beyond my wildest dreams. So I'm really pleased with what it's done, and there were some sort of core principles that I had when I started creating it. One is that it's community-based. If you want to participate and want to do something on the website, you're welcome to come and check it out. You're welcome to contribute code. You're welcome to talk to the folks that are also contributing code, or upload your images, things like that. Those are all sort of early ideas in this. And as Annie mentioned, I don't consider us in any real competition with INAT. I'm actually, I know the folks that work at INAT. Kenichi, who wrote it originally as a master's thesis while he was at the University of California, Berkeley, actually reached out to me when he was working on it for advice on how to do it, because I had Mushroom Observer running. So I sort of consider it a bit of a, I don't know, a stepchild or something like that. And I'm really, really excited about how far it's come. And the fact that it's, all of life. And the other thing that they've done that's very different from Mushroom Observer is they've really gone after serious funding. So they are currently housed as part of the California Academy of Sciences. They go for bigger grants with larger companies and corporations, or not corporations, countries, and things like that. They have people who are dedicated and working full time on this. They've put hundreds of times, if not thousands of times, as much money into their product as ours, and it's got a lot of polish to it, and it's got a lot more observations overall. But I do think that what John said, that we focus on mushrooms. Like, our thing is mushrooms and fungi. Our community is focused around that, and INAT is more dispersed. So you get a lot of information, but it tends to be people who are just looking at all kinds of stuff, and less, and the app itself is sort of less focused on fungi specifically. And like, we share data with them. We could share data if we found someone that wanted to grab all the data and stuff it into Mushroom Observer and figure that out. We just haven't ever really gotten to the point of doing that. But both of the projects are open, and we love sharing. So it is totally open source. It also uses open content licenses, and we're very open to change. Some quick stats. As of today, we had about we're getting close to 1.5 million images. We've got about 450,000 observations. We've got about 10,000 people who have made some reasonable contribution to the site. And so, like I said, this is beyond, I started out with 720 images that I just wanted to share with my friends, and it's turned into this, which is wonderful. So as you're probably well aware, it basically records, the who, what, where, when, images, notes about fungi. That's its main goal. Between the licensing and the data reuse policies that we have, the data does get shared pretty widely. This humble image of agaricus that I took in my front yard in LA has now become the representation of agaricus on Wikipedia. And pretty much, that's because, again, it was open, shared, and freely available. So it's kind of embarrassing to me at a certain level, but agaricus is the original genus, so it's like, that's pretty cool. So if you put your images up either here or INAT, they may well get used in broader areas and become part of sort of a cultural thing. So that's really exciting. For me, the most important thing are the people. And I think there's a really important quote from the open source community, which is, if you treat them as your most important asset, they will return the favor by becoming your most important asset. So, the people that are involved are wonderful to work with, and it is all volunteer. It's all just, people spending the time that they want to spend on it. It's not something that people's making a lot of money off of or anything like that, or any money off of. And it's just because we love mushrooms and want to share with each other and want to share with other people and have a real educational goal. And also, I believe in enjoying the struggle and encouraging the humor. So one of the things I'm going to give you is an early introduction to our mobile app. We are in beta testing for it. Annie apparently is one of our beta testers. Other folks could sign up for it, but we're kind of hopefully getting to the end of that process, and we'll soon have this available in the App Store and the iOS App Store and the Android App Store as well, whatever it's called. One of the exciting things here is we'd wanted to do mobile app for a long time, but none of the core developers had much experience with it. But it was the top request by the community, and a wonderful advocate who's part of our board of directors, Anne Pringle, had some friends at Google and talked to her friends at Google, and we ended up getting a small grant that has actually allowed us to hire a short-term contractor who has experience with the mobile app. I think she's also now become interested and addicted to mushrooms. And so she'll probably continue to be part of our community and continue to contribute in valuable ways. And we've been using that to encourage the development of this mobile app. There have been a number of sort of volunteer efforts to do mobile apps that have come up from time to time, and some of them have been reasonably successful, but none of them have really gotten off the ground. I think making this part of the sort of inner core of the app, of the development, will make a big difference, and we'll be able to keep it going and maintained. So one of the big goals with the mobile app, we've had a responsive app for a long time, so you've been able to look at the site on your phone. You've been able to do things on your phone as long as you're connected to the internet. The big problem is, a lot of times when you're out mushroom collecting, you're not connected to the internet. And so there wasn't a good way to record your information. You could take pictures and things like that and create it when you get back, but often you want to take the notes there in the field. You want to like do all your work while you're staring at the actual organism. So this does that. So we go in here, we click create observation, we can add some photos. I'm just pulling out of my gallery, but you could go straight from the camera, sort of typical mobile app type stuff. That uploads here. You can click the use info button here, and it will automatically look at the photo and the phone and figure out the location and the date from the photo. One of the things on the digital privacy thing that you should be aware of is that a lot of photos have location data embedded in them. And if you're not careful, your photos can share your location data that you didn't mean to. One of the things that we do in Mushroom Observer, as far as that goes, I don't know what they do on INAT, but we typically strip that information unless people give that permission. So there's this hide exact coordinates toggle, which you can turn on. If you do that, we will be sure to strip that information out of your photo, as well as making the location broad. And our locations are generally broad in our system. You can put exact Latin long, but when you put a named location in, it's just a rectangle. Like you'd put in something like Rock Creek Park, and it would be all of Rock Creek Park. So you found in Rock Creek Park, but you're not saying it was under this tree and under this rock or whatever. Go ahead here, you go ahead and you have an option to put in a name. You may not know what it is, so you don't have to put in a name. You do need to put some kind of location. If you really want to be obscure, you can just put Earth, because it was presumably found on Earth. But we encourage a closer location because, as we know, understanding what species you have, it depends a lot on the location. You can go in here, select locations. It has a list of locations that you can choose from. It does not currently infer your location based on the Latin long, which would be nice. But that's kind of, we decided for the first version, we weren't going to try to go there because it's a whole other service thing and it gets complicated. But anyway, we may well do that at some point in the future. So we go in here. For those of you who are familiar with Mushroom Observer, after we click next, we can go ahead, we can put in a confidence. So this is a set of confidence values that I put in at one point. It kind of needs to be adjusted, but it is something that I don't think INAT really deals with, which is the question of how confident you are in the particular ID and the ability for other people to say, yeah, I don't think it's that. They can confirm your ID, but that seems to be all I've seen. But maybe they do more. I don't know. Wouldn't surprise me if they do. I just don't know it. You're encouraged to put in some notes, particularly habitat's really useful. And then because we're out in the field in this imaginary scenario, we hit save and it just saves it in our draft area. And then once I get back and I'm doing internet stuff, I can connect in here, click on that draft, saying edit draft up there, and then I can click the upload button and boom, it shows up on Mushroom Observer. So that's the general purpose of the mobile app right now as it currently stands. And we think it'll be helpful for getting more observations and getting folks involved in doing things in the field. And Annie was saying that she'd worked on it and it's been working pretty well for her. It's been working reasonably well for me. We've got a few little bugs that we need to sort out, but we'll get those corralled soon. So that's the mobile app. So one of the big things I think is important about this, someone I think mentioned the dialogue that we have. We do have a kind of fun community. So this is an example of sort of more of a humor kind of observation here. So somebody put in Calvacia indigo here back. And this is actually, as you can probably tell, a deflated ball sitting out in the middle of a snowy field. But people had some fun with this, and this was also on the 4th of July, or on sorry, April 1st. It's just the 4th of July, but on April 1st. So we have fun and you know, play around with what we're doing as well as getting some serious mushroom and fungal research done. One of the things that John asked me to talk a little bit about was sort of the data mining / search stuff. It actually is quite similar to what you saw on INAT that Annie presented. The interface is, you know, a little older, a little more dated, but and it's sort of buried inside the search functionality. So if you go up to the search box and just type in something like Amanita, it'll give you a bunch of Amanitas. But you can also search by date and give a month. You can search by location, or you can put it all together. So as an example, if I just search for, like when I searched yesterday for Amanita, I got a bunch of results, but these were the top 3. One of them didn't even have an image, but that is supported in the system. Maybe you just saw it for whatever reason and wanted to report that you saw it. And then there are a couple other pictures with images. I can then reduce that or focus that down to Amanitas that are in USA, Washington, DC. Your location information is something that you have control of, or the order you put stuff in and location is controllable. But this is sort of the more scientific way of doing it. Most people do Washington, DC, USA, so that's the default. But if you flip it over to the other mode, you can do it this way. As you'll notice, these are 3 observations from Annie that are Amanitas that were found in the Washington, DC area. And as you can see, most of these, I think, are in Rock Creek Park. Yeah. So similarly, I can restrict to date, and this is similar to what you saw with the month search. So I can restrict to August, and this is August across all years, similar to what we just saw. So you'll see these 3 are all from 2001. It's a much longer list if you want to go and see all of them, but you'll notice that these are coming from Utah and Wisconsin and New Hampshire and so forth. Then you can combine it all together and just get in August in Washington, DC. And so here's some Amanitas that were found in August in Washington, DC. So our search criteria is actually, I think, significantly broader than what I've seen in the interface in INAT. INAT may well have more options that I'm just not familiar with, but like one of the things we can do is only show observations that have images. So the images : yes. So that got rid of that other one and brought in a different one. There's a bunch of other options. If you click this search help, it'll give you a list of all the different ways that you can do search in the search box. There's also an advanced search box, which is a little bit more of a filtery kind of thing, as was described in the INAT thing. It's not as beautiful because Kenichi's got a great eye for design and cares about that stuff more than the people who are doing our development do. So if you go to the advanced search, you can do things like find all the lichens with images and herbarium collections that are collected by a particular person. So these are all, Jason Hollinger is one of the other big developers on the project, and mostly focuses on lichens. So we do get good lichen IDs and so forth. But that's just another example of some of the data mining and search functionality that we have. Finally, I wanted to talk a little about image-based ID. So we didn't really see a demo of that at INAT, but they have quite an impressive image-based ID system, which is actually one of the things that got me thinking, taking it more seriously, I guess. For a long time, when people looked at our website, we'd get people say, hey, couldn't you just get computers to ID your mushroom photos? And back in the dim dark days of 2009 or 2010, that wasn't really a thing. And there wasn't really great technology for doing that and making it all happen. That has now evolved. And now we get, on a roughly weekly basis, hey, can we use your images for creating a machine learning model? Which is essentially the translation of this into more technical terminology that is now current and very popular, and is essentially the technology that INAT is using to do the image-based ID that they're doing. So obviously the thing I want to do is go from this image and get out something like Amanita merirubescens as the name of this thing I found down the street from me. So I personally have been kind of I had kind of not trusted the image-based ID for a long time. It wasn't really until INAT kind of made me wake up and realize that there was more potential for this than I had thought. I had a very similar opinion as expressed in this cartoon, which says, so this is your machine learning system. Yep, you pour the data into this big pile of linear algebra, then collect the answers on the other side. What if the answers are wrong? Just stir the pile until they start looking right. So it is approximate. but it really is pretty impressive how far you can take it. And it will never get to species for all species. Like there are just species that simply can't be identified solely based on a single image. And that's just reality. But what I do believe that you can do consistently is get down to a much smaller set of candidate species that you need to understand to get an idea or to try to better understand what your fungus is. So as I mentioned, iNaturalist has a pretty impressive system for doing this. When I threw this in that particular image, it came up with a number of names. The top name it gave was Amanita rubescens, which for those of you who don't know that, is a European species, which is closely related to Amera rubescens, but is not the same. It did not offer Amera rubescens as a choice. That is because it's a relatively recent name. But interestingly, if you explicitly say that's what it is, it does have a group registered in INAT under that name as a complex. I'm not quite sure why it's a complex in Amera rubescens, but I'm not quite sure exactly what the DNA is telling us about the rubescent species here. So probably a good question for Rod Tellis if you know him. He's been active on Mushroom Observer for many years as an Amanita expert. So Mushroom Observer does have, so again, we have these people who come in and say, hey, can we use your data? Well, some of them actually come back and say, hey, we have something that you could try using. And one of our developers, Jason, worked with one of them to actually produce functionality in Mushroom Observer that does do image-based ID. It doesn't have a very extensive model. It's not as detailed as the INAT model at this point, but it is something that we are working on improving and creating a pipeline for keeping up to date, which is kind of the challenge we've got right now. And I need to get my head more around it, that kind of thing. Because this was a researcher in Germany who just said, I'm going to go do this. And so we do have this. It's hidden as a beta feature. So you have to get special permission to see it because it's not very reliable yet. But it is in there. We do. And when I fed it this particular image, it actually successfully came up with Amera rubescens as the name. So it does a reasonable job given the choices that it has available to it. I similarly took Grifola frondosa, locally grown, hen of the woods, for those of you who don't speak Latin. Interestingly, I threw this at iNaturalist Seek, which is their app. It turns out the ID model on that is actually significantly older than the website and is not nearly as reliable, especially for fungi. They're hoping to update that, but if you do try to use Seek and it doesn't give you good results, try the website is one recommendation. But also be cautious using Seek because it will get things off and there are gaps in the information and so forth. Just be aware of that. So when I tried it with Seek, it just came over the Garricomyces for my Grifola, which was kind of disappointing because that's really pretty high up in the tree. When I tried it on INAT on the website, it did come up with Grifola frondosa as the name, but it also said they're not confident enough to make a recommendation. When we tried it on ours, we completely missed the ID and ended up coming up with Trametes versicolor and Burkandra adusta and some things like that. I guess, no, Grifola frondosa is third. Okay, so it did find it. If you notice that Trametes picture, it looks an awful lot like my Grifola picture. So I can kind of see what was going on there. And again, it's more about having enough data, enough training, doing it the right ways to get the data to work right. So what we wanted to go, so we did get Grifola frondosa out. I can't remember why I put, oh, this is a different image. That's right. I threw in a different image of an older, more clear picture. And Mushroom Observer did come up with Graphola frondosa as the ID for this one. INAT surprisingly called it Graphola spa, which is surprising because there aren't very many species in Graphola, and there certainly isn't any other one that is in Western or Eastern North America. So I don't know. Another interesting example was this Collibia luxurians that was growing down the street. I don't know how many of you are familiar with this. It's a fairly common thing that grows in bark mulch typically, and got renamed to Colibiopsis luxurians a few years ago, new genus. So Mushroom Observer like completely struck out on this, thought it was Armillaria tubescens. It probably didn't have Colibiopsis in the model, so it probably couldn't return that as a possibility, be my guess. INAT thought it was honey mushrooms, which is kind of funny because Armillaria tubescens is the ringless honey mushroom, so we're kind of saying the same thing. INAT did manage to find C. luxurians as a name, but it also threw out Pluteus pedestatus and Leucoagaricus americanus. Now, one of the things we do in our app is we give you a percentage confidence for the result, and we plan to make that data available. INAT does not do that. It'll give you a list of things that are presumably ordered based on the hit frequency or on where it lands. But it would, in my opinion, it would be nice if they actually gave you that data, but I can also kind of understand them like not wanting to complicate things. But anyway, Mushroom Observer tends to go like more technical in general, so and less shiny. So we do plan on making those percentages available once we have a decent model together. So sort of wrap that up. I'm sure I'm running over time, but in my view, image ID is promising, but one of the big challenges is around taxonomy. So I mentioned a few examples in here. There are some more complex examples that I chose not to go into in this talk, but I'm happy to talk over if people are curious. But try looking up Amanita gemata or taking a picture of like the Western gemmed Amanita, if you happen to have any, and throw that at INAT and see what it does. It's kind of entertaining. But the problem has to do with these species complexes, and INAT still doesn't do a great job of dealing with things that are recognized as distinct but do not yet have a really clear place in the scientific hierarchy. That's the problem with the Western Amanita gemata-like thing, or Western gemata-like species. Gemata is actually a European name. Nobody has been able to sort out what species actually occur on the West Coast and how many there are and things like that. So it's challenging. And one of the interesting things that I chose very early on to do with Mushroom Observer is to allow people to add names. So what has happened as a result of that is the community has come up with ways of naming things that we don't have good IDs for. So for instance, I think there is an Amanita gemata complex or something like that, that if you search for that, you'll find those Western gemmed Amanitas. That's not something, so INAT is very strict about where they get their names from, and they get them from, that gets into a whole complex problem, but they get their names from sort of a vetted source, but it also tends to be slower than Mushroom Observer, because like a new publication will come out and we'll immediately get it in. Somebody will have a picture of it and like get it into the system quite right away. And INAT will soon follow. Like it doesn't take very long to get it processed, but it's not quite as quick on the uptake. And especially if there's areas where there's a lot of debate, they'll often wait till the dust settles before they try to do anything. I do say in here, I believe that we need a formal non-Linnaean naming system. This is something I've written papers on and so forth. The most coherent example I can give you A quick example I can give you is this Russula cremora color. For those of you who haven't collected it, you can find a single individual, what appears to be a single individual organism, fruiting and producing both white fruiting bodies and red-capped fruiting bodies. They have shown that originally they had two separate names. Cremora color was for the white one, and Sylvicola was applied to the red one, but they are the same species. In fact, they can be the same individual. And the problem with this is obviously if you just have one of these and you throw it at one of these name-based or image-based systems, it's going to go different directions because there are a whole bunch of white Russulas and there are a whole bunch of red Russulas. And so in my view, we should just have a name for white Russulas and red Russulas, but that's not a taxonomically meaningful group because it's not, it's an observationally meaningful group, and it actually just better describes what the person actually observed. Because if I'm saying that this is Russula cremora color, I'm making some inferences and jumps there. It happens to be growing in a place where this is a common species, and there were two. So I have some other hints that are pushing me the direction to this name, but that's not always the case. And so in my view, it's much more important that we record what we observe and we have names that are based on observational characters, rather than jumping to the conclusion that this is the same species as some dried specimen that's buried in some herbarium that I can't even get genetics on. And you know, it's just the concept of a species is useful, but it actually doesn't completely map onto the observational experience. Anyway, enough on that. I'm happy to answer more questions about that or talk about it if people want to get into it. Finally, I just wanted to thank you all for your patience listening to me, and John for inviting me to be here. The Mushroom Observer community, none of this would have happened without more than me, hands on the keyboard. Jason Hollinger in particular has contributed a huge amount. Joe Cohen is a huge contributor. Alan Rockefeller, who also is very active on NINAT, is very active in Mushroom Observer and supports us with some of our image storage and things like that. And Pringle, I mentioned before, and many more. John is on our board of directors and really appreciate his contributions in that way. Most recently, I wanted to thank Alan Celestino, Jeff Simeon, and Olivia Hunter. They've all helped around the area of the ID and the mobile app. I also would like to do a shout out to Margaret McKinney, who I consider myself an intellectual descendant of. She was one of the first sort of amateur mycologists that kind of made a name for herself. She wrote a book long ago called The Savory Wild Mushroom. My grandmother happened to be her friend, and that's how I ended up indirectly getting involved in mushrooms. David Aurora for the Mushrooms Demystified book, which I read cover to cover multiple times as a child and got me to where I am. And also the Fungus Federation of Santa Cruz, which really solidified a lot of that knowledge, and many, many more people. And also I want to thank Cantharellus californicus, because it's one of the first mushrooms I found and got me excited about finding good things in the woods. So thank you all. Happy to answer any questions. Happy to hand it back to the hosts.
Yeah, so Claire is in the chat and is a data analytics master student. So she was jumping on this and being like, oh, this would be great for machine learning right before you said that it was. Which begs the question of if someone has skills in that realm of like the programming or the machine learning, are you all looking for people to actually get involved?
Absolutely, absolutely. If people are interested in that, even if you just want to like, you know, soak up a little bit of the knowledge, or if you like are an expert in it, we would love it. Like we're very open, very, you know, it's a friendly community of developers. You know, we have right now we're doing weekly meetings because we're trying to get the mobile app over the line. We're meeting on Saturdays to just like chat it over and figure things out. And we've been doing sort of bi-weekly meetings for a while, and just to understand where things are at. You know, Jason and Joe and I are all happy to help mentor people who want to get going. And there's a person who's really working on some new UI stuff, which I think is going to be really exciting. Like, we're actually going to get a reasonably up-to-date UI here one of these months or years, because we have someone that's really interested in doing all of that. And I can tell you all the technologies we're planning on using, but it probably wouldn't be that meaningful. But we are making progress there. We would love to have more people involved in the community. It's a lot of fun, and like I said, it's all open. It's all,
Yeah.
very, very easy.
How would someone get plugged into that? Should they?
You can. There's a webmaster request form on the website. You can post that, and hopefully one of us will pick it up. You're welcome to reach me. I can throw email address in here. You can send email there, or one of the mushroomobserver.org email addresses. If you don't hear anything, poke John, and John will poke me. So, But if you're interested, it would be great.
Yeah.
So, and we have had.
We may have found you a volunteer.
Okay. It was just coincidental that I had joined MAW in 2006, and that's when I started posting on Mushroom Observer. And back then, at least, and I don't post a lot myself. I'm only on the board because I'm a tax lawyer, and I was helping them on those issues. And I'm not very good on data analysis either. But it was a very friendly community, is what I liked about Mushroom Observer, because INAT was not there, and you don't get the kind of feedback on INAT that you get on Mushroom Observer. And I would get, text messages from, I'd post a silly picture of something, and I'd get a message with some intelligent commentary on it, and we could have a back and forth about it. And then I'd recognize these names. I say, "Oh man, this guy's famous!" And it struck me as a really friendly national community of people who were working on the identifications, and I really enjoyed it a lot.
Yeah, Rachel Courtney was asking a related question about, if you post a picture that doesn't have what it needs to have for someone to be able to make a good ID, will people comment back on you, or is there some more formal sort of feedback that you get? How does that work?
I think there's some comments in some of the docs, but who reads documentation?
Yeah.
People will often respond to it if they can, and say, "Hey, I like if I happen to see it, and I'll like put in a friendly note saying, I think it might be something like this, but I really need to see the underside to identify this, or if you could take the picture a little more clearly, that kind of thing." So we do, that's a natural thing I think that the community does, and we're happy to do that. So.
Annie's saying there are conversations in the comments of some observations on Mushroom Observer that are more educational than a whole chapter in a textbook.
Nathan, could you talk a little bit about the voting process and how it's always been a little bit confusing to me.
Yeah, it actually needs to be revised, and I can go and I've written, I know how I want to revise it, but I haven't ever gotten around to revising it, but I can describe what it does at the moment. When I originally wrote the app, I wanted to provide a way for people to weigh in on IDs and suggest IDs, and to be able to basically put in their opinion. And so I dreamed up this idea of a voting system where people could put in a value of how confident they are of a particular ID. So if someone posts something and they say, "I think it's this," that's fine. But somebody can come along and say, "Yeah, pretty unlikely that that's what it is. I think it's this instead." And that person who's making that additional assertion because they put doubt on the old ID and put some weight on their proposed ID will, at least for the short term, sort of win. They'll end up getting the ID on the observation and sort of the main, what we call a consensus ID. The other ID will remain, and some of what the little indicators say is whether a particular ID is agreed upon by the person who made the observation or not. Now, one of the interesting differences between INAT and Mushroom Observer is that, and I believe this is still the case, is that INAT will always show the ID that the person who created the observation applied to the observation. They will get suggestions from other people that they can then adopt, but at least at one point, I'm not certain this is still the case, but I think it's still the case, it will not change the name without the acknowledgement of the person who put in the ID. This was really important to Kenichi when he was creating it. I never fully understood why, because it seemed to me problematic in the long run, because if the person vanishes from the community, you don't want a bunch of things that are mislabeled. And so you can change it now, says Annie. Okay, you just have to get a 2/3 majority. Okay. Yeah, I'm not surprised they changed it. It was certainly a problem in the early days. And I know Scott Laurie, who's also involved in it, was kind of on my side of that conversation. And I guess finally convinced Kenichi that we need to be a little more, we need to go with the consensus a little bit more. So anyway, that's the rough idea behind the voting. I don't know how deep you want me to go into that, John. Is that enough, or do you want me to?
Do you rank the users, and so that some
Yes.
of their votes count more than other people's votes?
Yeah, so there's a contribution value for the site, which is sort of turns into a weight. Basically, you build up a contribution, and like the more committed you are to the community, the more you've contributed, the stronger your vote is. It's actually a logarithmic scale of that, so that, you know, people who are brand new get a little bit of weight and they can build up their weight a little faster. The people who've been there a long time, they keep getting incremental improvements, but it's not. So they don't just like completely overwhelm the voting. So, you know, the exact math of that I could go into. It's a little bit problematic, but it seems to do the right thing for the IDs that we do. The biggest problem in the existing ID system is that when I originally conceived of it, I expected people to generally vote, I'd call it that. That should, like, if that's the name that you would apply to it, I was expecting people would say they'd call it that. It turns out that there's actually sort of two dimensions of ID that I wasn't aware of at the time. One is what you think it is, and the other is how confident you are in that ID. And people blend this, and so the most popular thing to say is promising, which is the one down from, oh, I'd call it that, because people don't want to commit to it, or they feel like this isn't like quite accurate, or it's kind of like this weird like social thing. And the problem is that when you say promising, I can't tell in the algorithm, like in the way I put it together, are you saying there's some doubt of this, or are you doubting your ID in this? And the way I originally designed the system was that meant you had some doubt in it. So if you had a vote that was promising and a vote that was, I'd call it that, that actually was weaker than one person saying, I call it that, because someone had cast a little bit of doubt on it. Kind of makes sense, but it kind of doesn't make sense when you think about that, how that competes with it. What I've also realized is that, again, there's a little bit of stickiness in terms of the ID. We do some interesting things with synonyms and collating votes that are for the synonym of the same name and things like that. So that kind of gets around the renaming issue. but one of the challenges that we run into is that, or one of the things that's unfair, I guess, in the way it currently works, is that the person who's coming and doing the ID latest has the most information, and so should get extra weight because of that. Or really what I think should happen is the person who comes latest should be able to explicitly say, I really want to rename it to be this other thing. Because they have more information than the people who came before them. Now, you could get some bad behavior happening in there if somebody, for some reason, wants to misname mushrooms, but I think that's a really unlikely thing. And if it is, then we need to handle that in a separate channel than handling like how the information goes. And if you think about, if you look at how herbaria work, the ID on a particular collection in the herbarium is typically based on the latest notation that has gone on the herbarium collection. Now, the other thing that's kind of weird about herbarium collections is that they're sorted by the original name that they were put under. So the collector like puts some name on it and it gets sorted under that name, which is really difficult when you get taxonomic changes and stuff like that, because now it's like you have to look in 16 different places, but it also means that the curator of the herbarium doesn't have to move around all the boxes. Moving around boxes is not hard on computers. And so to me, it doesn't make sense to follow that convention, although we do have herbarium devotees who are aghast at the fact that we changed the name from what was the original observation name. But I don't think it's terribly useful to keep the original observation name because it loses value over time. So
my
So you've developed this. Oh, go ahead.
go ahead. My impression is that this is that the mushroom observer method, which allows for more uncertainty in expression rather than iNaturalist. You get a name and then you get two experts to say I agree, or you get two people who say I agree, and that's it. And you don't have a lot of discussion about that. So I think that's one of the advantages, that you have people laying out their analysis on Mushroom Observer, whereas you don't seem to see that very much on iNaturalist. And so you can learn a lot more, as Annie says.
So you have this huge database, and it's a great sort of community for us to get names put on our mushrooms and for us to store our observations and maybe do some data mining like Annie was talking about. And are there other uses? Are there other things that people are using the data that is in this amazing database for? Do you know about projects that people have used it for?
Yeah. So I mean, most of them are mushroom related. So there are people who come in and grab our data and do the image ID stuff. Like that's been a really popular use for our data, and we've actually done things to facilitate that in various ways, because it used to be a real suck from our database because people would pull stuff in a really slow way and it was problematic. But we made that a lot easier to do because we have a large number of people that are interested in the data set for that reason. We also get professional mycologists that are really interested in the distributional data and pulling observations to do studies. So the example is off the top of my head is Anne Pringle, who's on our board, but uses our data, is interested in the spread of the species Amanita tiersii, which is probably has not been recorded in Washington, D.C., at least not yet. But it's usually a southern and southeastern or southwestern southern species, and it is moving north. And it's probably a bit of an indication of climate change and some things like that. It's an interesting Amanita because it is I'm pretty sure it's saprotrophic and grows in lawns. It doesn't grow associated with trees. It's not mycorrhizal with trees. It's in that there's a group of Amanitas that do that. And it's really interesting to see the spread of that. And she's been pulling observations from Mushroom Observer as well as INAT to help her research of that. And I know there are a number, it's like when I go to professional mushroom events like there's a large number of people who like mention that, oh, I'm grabbing observations for this purpose or that purpose or the other purpose. And at one point I started collecting publications that were associated with it. And there's even a Mushroom Observer has an ability to add a publication. I think there's a publication tab over on the left-hand side. I don't think it's terribly active. I don't think people are being really great about keeping that up to date. But we were getting enough of them that, you know, and there have been a number of, like, one of the things, original hopes I had is that people would actually use it to help them name new species of fungi and that kind of, and that has definitely happened. There are a number of species that the publications thank the community, and you know, or they even got started with Mushroom Observer and got to the point where they were contributing to, actively contributing to mycological research. So that's really exciting to me as well. Our main target audience are folks like you all, people who are learning fungi and kind of stepping up to this, but we do get a fair amount of support from the professional community. And I consider it a complete win when someone like comes through Mushroom Observer, gets interested in fungi, and then ends up doing professional publication. They'll often like withdraw from the community to a certain extent at that point, because now they're like having to write grants and raise money and all that kind of stuff. So, and that's fine. That's just a total win to me, because the mycology community is a broad organization, has now got more people. So that's good.
This might be a question for Annie as much as for you, but what would you recommend as sort of a workflow if we're taking pictures and want to get them on both iNaturalist and Mushroom Observer? There's no way to like cross-reference or link that, right? Like you just have to.
There is.
Okay.
It is unfortunately at the moment only one way. It only goes from Mushroom Observer to iNat. iNat created an adapter where they can pull in observations from Mushroom Observer. For those of you who want to do programming and that kind of stuff, we actually have a pretty robust API on Mushroom Observer where you can grab stuff and download pretty much anything you want from our database. iNat wrote an adapter where they can pull stuff, they can pull user information out of there. Annie, I don't know if you're using that or how well that's working. Oh, we do have Jersey and DC. That's cool.
Oh, she linked to an iNat observation of it.
Oh, sweet.
Cool.
Nathan, does the uncertainty in identification affect the machine learning that you're doing, or have you gone through and vetted particular submissions or determined what the high quality submissions are in order to use that for the machine learning?
This is a really interesting question, actually. There are many layers to this question. So let's start with what iNat does, because it's the simplest to explain. They have a set of labels. So things like Amanita tiersii is an example of a label. And they have a set of observations that are, quote, the research grade observations that are associated with that name. And they take a set of names, set of images that are associated with that name. They actually, as part of machine learning, you have to split that set into a training set and a test set that you have to keep separate. Like it can be really problematic if you let those sets mix. So they have to separate those out into one set, training set and a test set. As you're building the machine model, it will associate that label with features that it finds in the training set. And then afterwards, it will look at the test set and say, did I get this right? Okay, so that's just like mechanics of how machine learning currently works. The challenge with that is that, one, if you've got, so computers are classic machines of garbage in, garbage out. Like that's like, if you put garbage, if you have bad IDs that are going in, it's going to have a worse data model. It just is. And if you don't have enough labels or you have the wrong labels, you will also create problems. So one of the interesting challenges to me is that we get a lot of input that can't be identified to species. One of our most widely used names is Agaricales, which is like the order. Like, yes, it's a mushroom, is basically what that's saying, which is interesting because there was something about that ID that, for something about that observation that wasn't sufficient to get any further than that, but you could create a label that was Agaricales and associate those things that end up in that bin with that name, and thereby determine essentially this is a bad image for getting to species level. Similarly, we get a lot of images that are microscopy, and it would be nice to be able to identify microscopy pictures. It's unlikely that you're going to be able to identify to species with microscopy images, but you could put them in a bin that was microscopy, or label microscopy, and kind of divide it up that way. And so one of the things I'm thinking about in how I want to go after doing image ID is to look at the stream of images that come in and where they get categorized, rather than taking the perspective like it's gotta be something that we already know the name of, and use that as our test set. So if you have like the last 10,000 images that came, the last 10,000 observations that came in, you could look at all the labels that were applied to those and then use that as your training set, rather than take all the things that have been identified to species and are scientifically like research grade, and use those. So it's kind of a different way of looking at it. And I think it actually is more the ID process that actually happens, and has the ability to potentially do things like say, like it could almost in an automated way say, this observation isn't right to do species level ID, and maybe give you some instructions on like, this is how to take a better picture. Like it could actually identify like, that's just a blurry picture of the top of a mushroom. You need to show us the underside. Like we could actually get to that point. Now, the interesting thing is INAT can kind of never do that. Well, I never say never. Who knows what they're going to get going in the future. But if you think about INAT's remit, it's all of life. And if you look, and you can't say, show me the underside of a whale, right? Like that wouldn't be what you want to do, right? And so like they have this problem like at many different layers for the different groups that they're looking at. Whereas with mushrooms, like the requirements for taking a good photo of mushrooms, you can get a lot more specific about how to take a good picture than you can with all of life. So anyway, few of my ramblings.
Yeah, it's fascinating. One thing I've noticed with iNaturalist too is that the AI is looking picture by picture. So if you like scroll through the 4 pictures that you uploaded for an observation, the suggested IDs might change.
Mm-hmm.
Yep. And is that something, and that must be something you're having to think about too, of like, do you take all of the pictures for an observation into account at once somehow?
Well, the interesting thing is the underlying technology is really based on single images.
Yeah.
So combining results like it is something I've thought about, but combining results like you're talking about is a little tricky. It's not impossible. And it kind of ties into something else, which I think is really interesting. So you can get results for each of those 4. And if you know they're the same observation, you can use those results, like you can compare and contrast those results.
Right.
So like if Amanita tiersii is 1, 3, 2, and 3 in your IDs there, and there's no other name that ranks as high as that, you have increased confidence that that's Amanita tiersii, right? And you could do that calculation and express it. one of the interesting questions that you run into is what happens if the correct name doesn't show up in one of those observations because you took a picture of the surrounding habitat, right?
Mm-hmm.
And like the actual organism, like maybe it's there, but it's like a teeny white dot down in the bottom, of a big grassy field, right? And it's identifying it as this is a grassy field, right? Which is accurate,
Right.
but so there's some interesting challenges there. But one of the things I've thought a lot about is there are observational sister species. So a great example for me is the Krugonthus species. I don't know how familiar you are with those, but it's also known as the pine spike. So there's Krugonthus vinicolor, there's Krugonthus rudilus, and there's Krugonthus ocratius. Rudilus and vinicolor cannot be accurately distinguished without a microscope. Like, you just can't do it. There are people who claim that there's like certain habitat clues and some things like that. Maybe that's true. I've not seen published research that actually supports that, but people have like that feeling. Okay, maybe. But if you're just looking at the image, like you just can't get there. One of the things that I think you could do with a machine learning system is look at those IDs that often come up together, right? And circumscribe essentially around that set of names that are always coming up at the same time, right? Because like these are ones that are really hard to distinguish. And this is where, in my opinion, you should create one of these non-Linnaean formal names, right? Like these are, smooth pine spikes, right? As opposed to Krugonthus tomentosus, which is the hairy pine spike, which you can actually distinguish reasonably. And you might be able to keep out of that set. But you could create these circumscribed groups that you then describe a set of species that are associated with. And when you find those circumcised groups, you can put in information about like, okay, you're in this group. That's great. We've narrowed down the set to 5 or 10 or 15 species. Here's a key for how to distinguish them, or here are the properties that you need to look at to make this distinction between these species. And in my opinion, this problem is only going to get harder as we get more and more genetic information. Because, for example, if you look at like Armillaria, the honey mushrooms, there are 14 species of Armillaria that occur in North America. Most people will just all call them Armillaria mellia, right? But that's not right. They should all be honey mushrooms based on the observational characters. There are a few you can distinguish based on, are they growing in clumps, or are they, like there's a lot of details you can get into about how to distinguish those, but not all of them are actually visually distinct enough that you would ever have any hope of distinguishing them using an image-based system. So we've got to have a name for that result that then tells you, and here are the different kinds. You may be able to do some of it regionally or some things like that. But anyway, it gets into some interesting complexities there that I definitely want to dig into and understand better.
Yeah, that's fascinating. And with a relatively circumscribed group that's just fungi, you can probably do that more easily than an organization like INAT can, where they're dealing with everything.
That's my hope. And I think not only do you have more hope of doing that with fungi, I think there's also more reason to do it with fungi.
Yeah, right.
Like if you compare it with something like birds or flowers or trees, like which is the vast majority of what goes into INAT. Like the vast majority of observations, the vast majority of names are animals and plants. Not surprising, like of course. And it's far easier, and you have less of those issues in there. The problem you get into with plants is hybrids, which is a whole other problem that INAT also calls out in some of their discussion about how to do image-based ID. And they basically throw up their hands at hybrids right now. They're hoping to get there, but that's a really hard problem. Thank God we don't have to worry about that in fungi. But at least we don't think we do.
Do we even know? Right.
We don't even know.
So Douglas Miller, I think, is the person you called on at the very beginning of your presentation because he had his hand up and his hand is still up. And Douglas, if you are out there and if you actually have a question, feel free to jump in. And if folks have other questions, we're down to a small enough number of people. I think if folks want to jump in, that's fine. But we've already answered.
Do they have the ability to turn on their mic? Does Suzanne have a question?
Yeah, well, it's sort of a question. I'm a complete new person. And so, and
Wow.
although I did volunteer at the folk thing, and that was really exciting because everybody who came up to the table had some fascinating new take on things. But anyway, so I'm in the lower part of Howard County by Savage Park. And so just to do my own initial foray there, I could map out what I'm looking for by going to the iNaturalist and seeing what is there, what people have reported finding in the vicinity in June or July in previous years. Does that sound like a sort of how to get started in where to look? I mean, I can just go out looking, but I mean, if I want to replicate other people's findings, so to speak.
I mean,
Does that
in that
sound like-
sense, it's a good way to sort of prime yourself for like, okay,
Right.
these are the things that I
Yeah.
might see. I should be looking for this kind of stuff. These are the names that maybe will come into my brain when I look at the thing in the field, and maybe not. Maybe I'll have to go look it up again. But yeah,
can definitely-
so much of it is just repetition, right? And like seeing the things over and over until you remember what they are.
Yeah, you can
Yeah.
also look at Mushroom Observer too. Like I've created, and I know other people have created locations like Martin Livesey has created amazing Rock Creek Regional Park database of observations. I have a pretty large database of Rock Creek Park, and most of the other parks too, if it wasn't already created by someone, we've created a location for it. Like I've created locations for Scott's Run, for some of the other local, a bunch of the other local parks like Great Falls, Maryland, Great Falls, Virginia. So if you also want to, like, I use both a lot, and I think they're very complementary of one another. So whatever you feel like looking at, you can either kind of search in a area that you create, or you can search for a specific park or location.
Okay,
And
thank you. But it is kind of neat. No, all the stuff there that helps you feel like you're not just frittering your time away taking pictures of things, which is what I've been doing.
So one of the things I didn't show in my demo, but which works in Mushroom Observer as well, once you do a search like I described, where you search for a region, there's a link in the upper right hand corner which says show map or something like that. There's a maps button there, and you can click on that and it'll lay it out on a map and show you where the observations were. So that can
Okay.
also be helpful for people who are more visually oriented.
I know,
It
so
uses the
yeah, I'm also a birder, and so there's a whole nother sort of community science thing called eBird.
Oh,
that is-
the wingspan, is
Right?
that wingspan?
yeah.
Yeah, it's funny because I know somebody who, we have a common friend, me and Elizabeth Hargrave. It's someone who does wingspan.
Great.
So
But like one of the biggest things that I do with eBird is just look for the concentration of all observations of birds in different places, right? And I imagine you could do that with fungi too. So instead of looking for like what specific species are people finding in different places, just like how many dots are there on the map in different places? And part of that is going to be, where do the MAW members live and are reporting lots of mushrooms, right? But it's like that can be like, where is
Right.
it worth going?
like you're getting
People
warmer.
A lot of
you're
like
getting
maybe it's a
colder.
moister location for some reason, or it's got nice old trees, so there's lots of mycorrhizal mushrooms in there. Like you can see sort of a heat map of where is it worth looking for stuff,
Right. Okay.
whether
Well,
you
I
care about
appreciate
specific
that.
species or not.
It's also worth keeping in mind that more dots are going to appear where there are more people,
Yeah.
because people are going to tend to go to places that are nearby. So there might be tons of mushrooms in a place 2 hours away, but relatively few dots on the map, right? So you've got to take that into account too.
Right, for
Yeah, I mean-
MAW members who are interested in following up on AI identification, there was a consortium program by Henry Beaker who did I guess the narrower you go, the better you can do. He did a worldwide project on hebeloma, and has just an incredible database, and all different ways of sorting it. Basically, you put in different characteristics down to microscopic characteristics and find the probable identifications. And that program is available. The link to the YouTube video of that is available to our members on our website under the consortium presentations.
Yeah, so Nathan's sharing his screen and showing sort of the heat map of DC, and I think none of us would be surprised about the concentration of stuff in Rock Creek Park, but what is going on at the Supreme Court? Like all those dots to the right of the Capitol.
Oh, yeah. What is
The
there?
mushroom hotspot. Oh, they're a little more spread out. Okay, but there is one right
Too
on the
much-
Supreme Court.
Too much old growth.
Right by Union Station, there's a little park with some old trees in it.
Yeah, I mean, the Capitol has beautiful old trees in their grounds too, so yeah.
Right, and there was the IRS building was kind of right across the street there. I went for a meeting there once and pointed out to the guy I was meeting with that there was some honey mushrooms that were good eating right down in front of the planter in front of the building. It was right by Union Station. He probably thought I was trying to poison him.
Yeah.
Well, thank you,
Well, it's
Nathan.
like
That was
someone
great.
found some agrosapi, a Clibiopsis, and
Yeah,
Ganoderma.
right.
So,
And it's probably the
yeah.
kind of thing also where if you find something growing on the grounds of the Supreme Court, you report it because you're amused by the fact that it was on the grounds of the Supreme Court.
Yeah.
Oh, that's fun.
Great.
Well, this has been great. We're running past 9 o'clock and I want to let everyone get going. But thank you so much, Nathan, for your time, and for everyone.
Oh, thank you. And it was great meeting you all. And, it'd be fun to try to get to one of your events.